standard way of collecting microbiological sample from the soil, which I will start with introduction. The soil, I will start with introduction of the soil. The soil is a natural occurring mixture of a mineral, organic, water, and micro or living organism. So now, how to collect a standard microbiological sample from the soil? It involves different steps, which to enable or to ensure the sample are representative and free from contamination. So the first step is preparation. The preparation the first preparation is to define the sampling objectives, which is in such a way you target your analyzing microbial community, detecting of pathogens, and assessing soil fertility. So the second, based on the preparation, because we have different types of preparation, when you want to collect the soil sample, you will choose the right sampling tools. You choose the right sampling tools, selecting a sterile and durable equipment such as shovel and some other sampling tool. The third step, when you are preparing for the collection of the soil sample, you will consider the preparation of the sampling container. You make sure that the container is sterile before you collect the sample by using different sterilization methods. So wearing of protection equipment also, like wearing mask protection and to prevent exposure to potential pathogens. So also part of the preparation when you want to collect the sample, you have to have a notebook for the feed notes and pen and marker for labeling after the collection of the sample. The second step for the collection of the soil sample, you have to take note of the site assessment. When you are talking about the site assessment, you have to identify the area of interest based on the research objective. Secondly, you have to observe the site for the variation in soil type, the moisture, the vegetation, and the human activities. Based on the site assessment, you have to take note of choosing a multiple sample location if the area is heterogeneous. When you are preparing of collecting a soil sample, you have to consider the collection methods, which is the third step. So the soil depth, you have to consider that when you want to collect on the collection method, you have to consider the sample depth. You have to consider the sample size of the soil and the sample frequency is part of what you have to consider. The first step, which is handling samples, how you handle your sample when you want to collect the soil sample. You have to place the collected soil in a sterile container immediately. And also you avoid touching the inside of the sample container to avoid the contamination. So, and the fifth step, you have to label after the collection. You have to label by giving the sample names, like sample ID, the date, the time of collection, the location, details. You have to consider that. So the 6 steps, when you want to collect the soil sample, you have to take note of the storage and the transportation. And the last, which is the documentation, you have to record all the relevant information in the notebook, including the sample location description, the environmental condition, which involve the temperature, the moisture, and any other observation during the sampling. So that's just the summary of how we can collect a microbiological soil sample. Thank you for listening.
Did you mention depth? How deep should we go when we are taking sample? Did you mention how deep we can go when we are taking soil sample from the soil?
It depends on the literature or the instruction by the supervisor or the literature review you want to use. But commonly, if you want to take note of, if you want to take the sample from 0 to 10 centimeter, which represents surface of the soil, and 10 to 20 centimeter represents the subsurface of the soil.
Yes. Did you, when you are doing your